Welcome, everyone. Thank you for joining us for today's webinar, Biomarkers of Aging, presented by Dr. Alex Zhavoronkov, founder and CEO at Insilico Medicine. My name is Sarvi Greneman, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Zhavoronkov. I will now turn the presentation over to him.
Hello, ladies and gentlemen. It's a great pleasure for me to present to you today via LabRoots. It's my first attempt at this particular presentation style, and please forgive me if I kind of steer or click the wrong button here. I'm presenting as the CEO and founder of a company called Insilico Medicine. We are the next-gen AI company focusing on drug discovery, biomarker development, and aging research. We are the CB Insights Top 100 AI companies in 2018. We are Frost & Sullivan Best Practices North American Artificial Intelligence for Aging Research and Drug Development company this year. And we are on pretty much every analytical report. We publish at a rate of about 2 research papers a month, pretty much more than most AI companies out there. And we'd like to share our work in biomarkers of aging today, even though we focus on the entire full spectrum of pharmaceutical R&D and try to generalize in multiple areas. Multiple disease areas, and also in multiple phases of pharma R&D. So first things first, one of the most inefficient industries on the planet is the pharmaceutical industry. If you think about what industry is going to be the most disrupted by AI going forward, it is likely not going to be the transportation industry, because in transport, in automotive, you pretty much get from point A to point B 99.999% of the time. You can only make this process more efficient and reduce the number of accidents. In the pharmaceutical industry, you've got 92% failure rate, usually after the molecules have been tested in mice. So those failure rates translate into enormous costs, an enormous cost or R&D burden for the pharmaceutical companies. And it costs more than $2.6 billion to develop a drug from preclinical to market stages. So this slides depict how the pharmaceutical R&D looks like. So those gray dots represent the kind of number of failures per phase. So there are lots and lots of compounds that you usually start with in the basic research stage, progress into drug discovery, preclinical phase. And then there is a large number of failures in phase 1, phase 2, phase 3. And then if you fail at the FDA review or phase 3, it's usually a very expensive failure. So the goal of our company, and I think of the entire AI for pharma industry today, is to develop really great molecules for precise right molecular targets delivered at the right time to the patient at the very early basic research stage, to reduce the failures in the various phases of R&D. And that's our core goal. To do that, you need to focus on multiple areas of pharmaceutical drug discovery to make sure that you predict everything in the future of the pharmaceutical R&D cycle to avoid all kinds of expensive failures. And at Insilico, we offer a very unique blend of biology, chemistry, and digital medicine expertise, and try to train our models to fit the biological hypothesis, chemistry knowledge, chemistry expertise, and also looking into the future into digital medicine. And we have a set of tools for that. So this is how the pharmaceutical drug discovery pipeline looks like. Usually you have a hypothesis and knowledge discovery phase where you identify the disease target association and also hypothesize about the origin and etymology of the disease. Then you progress into identifying specific biological targets that drive the disease or that are causal or that are passengers in the disease. But suppressing them or activating them might be beneficial for the patient. Then for those specific protein targets or sets of targets for a specific condition, you start generating specific compounds, and we are one of the big players in that space. You can do it via virtual screening. So by screening known chemical space and known chemical libraries against that protein target, or you can do what we are trying to do, and I'll briefly touch on that. You can generate novel compounds for a specific target. Then there is a process for ADME tox prediction. There is clinical trials, so all those phases where you actually go into humans. Then it's a good idea to personalize your molecule for a specific patient subpopulation, so you're addressing only those patients that are most likely to respond to the treatment. And it's very important to collect real-world evidence and real-world insights into a system which can also relate back into hypothesis and assist the company or the entire industry with new insights coming from patient populations. And of course, you can use AI for marketing of the products, because if you know the patients that are likely to respond to the treatment and most likely to benefit, you can cater to those specific patients through the doctors or through the various organizations that deal with those patients. This is how our modus operandi, kind of the central dogma of drug discovery using AI at Insilico looks like. So we focus primarily on identifying protein targets within a variety of diseases, primarily using transcriptomics, proteomics, and metabolomics data coming from public and also from our own databases. We try to establish this disease target association to link the right target or sets of targets to a specific disease. And we go into prior art and look at grants, publications, patents, clinical trials, and also many other sources to strengthen our hypothesis for disease target association. And once we establish a little bit more evidence and stronger signals for a specific protein target, then we go and generate a specific novel molecule or a set of molecules for that protein target. Then we run it through sets of predictors, predictors of clinical trials outcomes. Then we send the molecules and actually the structures to our partners that synthesize those molecules. Currently, we work with WuXi AppTec. It's kind of the Ferrari of synthetic chemistry that manages to synthesize molecules very, very quickly, very precisely, and also captures a lot of valuable data that they pass back to us after we contract them to do that. So highly recommend that particular partner for synthetic chemistry and for many other preclinical exercises. So after you get those molecules validated, we license or partner on highly promising hits and leads and take a small percentage in the molecule when we license it to a smaller biotech or even to big pharma. This is a little bit more expanded view of what we do. Again, most of the clinical trials fail because you really miss the target in a specific disease or select the wrong patient subpopulation for those targets. But we, of course, link it to chemistry and try to link chemistry back into biology to assist target ID process with the chemistry expertise and also work in clinical trials outcomes predictions. We publish some of those pieces in peer-reviewed journals to help other academics and industry partners to get into those fields and to replicate our work. One of the hypotheses that I came up with at Insilico is that the various diseases have their own clocks. So you really need to capture, not only need to stratify the patients into specific patient subpopulations and identify the right patient for the right drug, but also the various diseases, once they manifest into a set of symptoms, they become pretty much irreversible. So you start treating the symptoms, but it's very difficult to go back to the root cause. Some of the examples are, for example, scleroderma, which starts as an autoimmune disease and manifests as fibrosis, sclerosis, and many other symptoms. You really need to fix those symptoms first, the manifestations of the disease on a tissue-specific level, on a systemic level, before going to the root cause. Fixing the immune system might not be enough. So you really need to also time the intervention to the specific disease clock. And to understand that particular disease clock, we need to understand biology in a very holistic manner. And one of the areas that allows you to look at biology in a very holistic manner on a tissue-specific level, on a systemic level, even on the population level and psychological level, is aging research. And one of the kind of simple ways to describe what happens with us during our life is this slide. So we are born into this world, we grow, we mature, we reproduce, we peak as individuals, we take care of our young, and then we continuously decline, get sick, and eventually die. So with prevention and with diet and exercise, you can shift this curve to the right, but the real outcome and the end outcome is pretty imminent, and it's very difficult to do something about it, and also to protect the individual from diseases in the later years. So the only way to prevent that is by proactive intervention and by really understanding the aging process and ways to rewind or put a stop to some of the aging clocks. So this is kind of also a philosophical chart, but also it might help you think of the meaning of life a little bit differently and help us become better doctors and better scientists. So there are multiple ways to measure human performance during our lives. So one measure is money, net worth. There is also reputation, number of kids, level of happiness, how we live, so number of experiences, all kinds of skills we aggregate, physical performance, how good of an athlete you are for some time. Again, there is a clock that's ticking that affects that particular feature and the score, years of life left, technological advances that we bring to the world, number of connections that we make, the resources that we accumulate or create, all kinds of power functions. And there is another score that I myself personally optimize for and the company is optimizing for, and that term is QALY, Quality Adjusted Life Years. QALY is a very well-known term in healthcare economics, and that's basically a year of life lived in the optimal state of health. Basically, if you are to be sick and be maybe half of your physical performance, operate at half of your optimal abilities, that would be half of the QALY. So to get one QALY, you need to live for 2 years in that particular state of physical activity, physical performance. So one QALY is one quality adjusted life year, a year of life lived in the optimal physical performance. So at Insilico, we are trying to maximize the number of global QALYs, and it's a very simple objective function for the company, for the individual, and for our algorithms. So we try to maximize QALYs for the individual, so stretch the life in optimal health for as long as possible, and also treat aging as a staged disease. And what it turned out to be, aging research turned out to be a very, very good mechanism for making AI more interpretable. So when you're training deep neural networks on continuous longitudinal data annotated with age and comprising of very simple sets of features, we can look at what kind of features are more important for making a prediction of the individual age or biological age or chronological age, and building causal graphs, looking at how the importance of every feature changes in time, giving us some idea of how the biology changes in time, how damage accumulates. And it turned out that AI can be a great tool for aging research as well. So it gives you a very holistic way of looking at things, looking at biology. As well, focusing on aging research, and there has been many studies done on that, performed on that, is probably the most impactful area for increasing life expectancy for the populations in the developed countries and also in developing countries as well. So if we cure cancer nowadays, it's going to add just a few years to life expectancies in developed countries. If we delay aging and slow down the age-associated retardation, we can gain decades in a healthy, productive life. As well, aging, as all of us know, and it's a statistically proven fact, is very related to disease. Most of the diseases are age-related. We accumulate damage over time, and that damage helps us develop all kinds of diseases, and at some point of time it breaks into processes that are irreversible. So when you're looking at many cancers, for example, or many other diseases, and look at them in a disconnected fashion, so look at many patients with various heterogeneous cancers, you're looking at pieces of the broken glass and trying to piece them all together, or try to find spots where we can intervene and glue them back together, or completely eliminate those pieces of the broken glass to ensure that the rest of the kind of glass can be maintained and remain unaffected. But aging research allows you to look at some of those processes in time and look at how this glass accumulates damage and breaks into disease. So many data sets out there that lack annotation, biological annotation, or have a specific sets of parameters for a specific disease usually do have age. So age is the most abundant feature out there. Everybody has age. Not everybody has diabetes or cancer. Every cancer is different, but everybody, every living creature, and actually everything has age in this world. So if we train deep neural networks to predict age on multiple levels and on multiple data types, it gives us a very holistic view on biology, and we can then retrain those networks on specific diseases using a process and methodology called transfer learning or one-shot learning. You train on age, and then you retrain on diseases. And we can also look at what specific features are most important in all kinds of biological processes, but primarily looking at aging. And at Insilico, as I've mentioned, we do publish consistently. We have an academic culture. And this is a list of some of our publications related to deep learning. And some of our publications that I'm going to cover today are specifically focused on deep biomarkers of aging. So deep neural networks trained to predict the age of the patient and either retrained on diseases or used to identify biological targets or used to develop new methods for interpreting biological deep neural networks trained on biological data. And one of the recent papers that we've published in Aging Research Reviews, one of the high-impact journals in the field, is this. So it's called Artificial Intelligence for Aging and Longevity Research on Recent Advances and Perspectives. And it covers quite a bit of our aging clocks that were developed by our group and also other groups in the field. And it touches on multiple applications of AI for drug discovery, biomarker development, and aging research. So you can use deep neural networks for building aging clocks. So I'm going to cover this in this presentation. But you can use deep learning also to identify geroprotectors, molecules that either mimic certain biological processes, for example, caloric restriction, or the activity of pharmaceutical compounds like rapamycin or metformin. Or you can look for molecules that negate the aging signatures in a variety of tissues, kill senescent cells, or target specific aging mechanisms. And that particular paper has an overview of that. You can use deep neural networks for biomarker development, including extracting valuable biological pathways from deep neural networks. You can use AI for compound similarity scoring, cross-species analysis, target identification, etc. So as I've mentioned, one of the ways to apply deep learning for aging research is to train deep neural networks to predict just one feature, age, using multiple data types, and also coming from patients of various ages, ideally from the same patient over time. But we can also glue together multiple data types just by training deep neural networks to predict age, and also identify multiple biological features that are extremely important in a variety of biological processes from deep neural networks trained on those various data types. And also understand what data type is more valuable for your particular application. From those deep neural nets trained on age, we can extract relevant biological pathways, identify targets, draw causal graphs, and also use those deep neural networks for synthesizing new data previously that does not exist in nature, just by shuffling this particular feature, age. And I'll illustrate that. So here we are using a technique called generative adversarial network, where two deep neural networks are competing with each other to essentially imagine novel objects or novel data that does not exist in nature with specific generation conditions that we specify up front. And here we specify just the age of the person. So that's myself. I am 40 today. Here, I actually think I do look 20. That's a computer-generated image. And here, we didn't do anything with the hair, but you can see that the deep neural network really modified my face to look 70. And this is just a graphic example of what happens when we apply generative adversarial networks to all kinds of data just by manipulating one feature, age. When we do it on other data types, like, for example, transcriptomic data type or blood tests or proteomic data or epigenetic data, the deep neural network does much better than with images, because here you've got a lot of features, basically hundreds of thousands or even millions of pixels. When you're doing it with transcriptomic data, you're working with 20 expression levels of 20,000 genes, or if you're working with blood tests, you might be working with just tens of features that are reconstructed using GANs. And you can very quickly check whether the data synthesized using AI is good or not. In our case, we did test this data, and many experts fail to distinguish between synthetic data and the data generated using our GANs. That's how good it is. You can now synthesize synthetic patients. You can synthesize patients of a variety of ages and also with a variety of conditions. You can introduce a mutation, for example, into the transcriptome, for example, and see how that mutation affects the transcriptome going forward of a patient of a different age. And most importantly, synthetic data generated using those GANs. Currently, that's kind of up for discussion, but it doesn't seem to belong to a specific patient. So it's computer-generated data synthesized from millions and millions and millions of features obtained from consented data, and we can now generate virtual populations of human patients or animal patients or animal models. You can also do the exactly same thing with cells on a tissue-specific level or a culture-specific level when you're working with cell lines, or you can even do it on a single-cell level. Again, train the deep neural nets to predict the state, the differentiation state of the cells, or the aging, senescence state of the cell on multiple data types. And then again, extract valuable pathways, targets, causal graphs, generate synthetic data, synthetic cell data. You can also screen for geropredictors. So for example, when you're working with transcriptomic profiles, you can compare the transcriptomic profiles of young patients with the transcriptomic profiles of old patients and look at what features change during aging and what features can be causal and what features can be targeted, and identify molecules that can target those specific features. Or in one of the studies, we looked at specific a range of molecules that induce certain transcriptional response, linked it to mesh terms, reduce dimensionality using signaling pathway analysis, and train the deep neural networks to identify whether this molecule might have an anti-aging and pro-longevity activity or not. And one of the tools we developed for working with transcriptomic data and also proteomic data, and actually many types of omics data types, is called IPANDA. So this technique allows you to group genes into networks and look at the differential state change of those networks in time or between different states, like, for example, normal tissue and tissue afflicted by disease. And that algorithm is used ubiquitously in academia and industry. We use this tool to go down from 20,000 genes to maybe 200, 300 pathway scores and train deep neural networks on lower dimensionality data. And also those scores are usually much more robust and much less susceptible to variability induced by batch effects or experimental conditions or experimentor introduced conditions. And one idea we got a few years ago in 2015, again, our company started in deep learning in 2014 and in aging research way before that. So one of the ideas was to create a data set of young and old patients. So basically gene expression data annotated with age, and build aging-specific signatures using this IPANDA algorithm by comparing old cells and young cells, and then looking at transcriptional response experiments like the Broad Institute's connectivity map or LINCS, where a very large number of cells and cell types were incubated with a large number of compounds. and gene expression was measured before and after incubation with a compound, and build kind of compound-specific response signatures in a variety of cell types, and then see if we can negate those aging signatures with those compound signatures, compound-induced signatures. So and then we got an idea that why don't we identify natural compounds targeting senescence pathways using that particular technique, then launch combinations of these compounds as nutraceuticals, and then measure a variety of biomarkers in real world and evaluate the possible side effects. So people take those nutraceuticals anyway in millions, so why don't we also try to measure the effects? So we published a couple of papers on that in credible academic journals, peer-reviewed academic journals, did some experimental validation on a cellular level, and then partnered with a nutraceutical company called Life Extension that caters to about 450,000 people, predominantly in the United States, through a catalog. And those the users of Life Extension products are usually a little bit more educated than the general public that is taking nutraceuticals. And Life Extension is offering annual blood tests for their user base, or regular blood tests with a partnership with LabCorp. So you can actually take a blood test and see whether a specific nutraceutical is working or not. It's very difficult to identify whether it's working or not. So we launched a series of nutraceutical products, primarily comprised of the molecules that people are taking anyway in large quantities for anti-aging. But we have specific rationale for those molecules to be consumed in those combinations. And we tested some of those molecules in vitro. It takes us about 4 weeks to predict, 25 weeks to do safety testing, also ensure that it's GRAS approved, and combined in a specific capsule. Then the product is launched, and then we expect people to upload some of their data in an anonymous way. It's totally anonymous through a system called Young.AI, and then we can test and analyze. So currently we're collecting those data types through a system called Young.AI. It's currently in beta. And that system allows you to upload your photos, your blood tests, wearable data, ECG, and transcriptomic data. We're adding microbiomic aging clock soon, and then see how a variety of interventions might affect your predicted age. And this is how the dashboard looks like. And now we're getting into the kind of topic of the aging clocks and aging biomarkers. So this industry has been revolutionized by Stephen Horvath's work. Steve Horvath out of UCLA in 2013, who published the first aging clock, epigenetic aging clock in 2013 on genome biology. I highly recommend looking at his paper. Huge respect for this work, scrupulous and single author publication, showing that you can now use methylation data to predict a person's age with 2.7 mean absolute error. So it's basically plus minus 2.7 years for guessing your chronological age with reasonable consistency. So it's a very robust clock. There are other epigenetic aging clocks, so pretty much every group developed their own, and including ourselves, using the various techniques, including deep learning. We also published the first biochemistry-based clock using deep neural nets, which is about 5.5 years accurate. It's not as consistent as epigenetic aging clock. There are multiple other clocks, like transcriptomic aging clock, telomere clock. So at Insilico, we have 16 clocks to date, developed to date on the various data types. And you can develop those clocks using a variety of methods. So Stephen Horvath's aging clock was developed using ElasticNet. Hannum's clock, ElasticNet. Then our clock was developed using deep neural networks. And you can even develop aging clocks using linear regression when you're looking at the length of the telomeres. It's actually one of the worst clocks out there. Also, in my opinion, one of the least biologically relevant. So one of the ideas of developing aging clocks is to train a feed-forward deep neural net with multiple features, for example, regular results from regular blood tests as input features into the deep neural net, basically one feature per neuron. And you can use various techniques to evaluate the, so like deep fish selection, for example, to evaluate the net contribution, the contribution to the accuracy of the predictor for every individual element going into the deep neural net by developing single connected layers of neurons. You've got an input layer where you know each feature that is going in, and you've got weighted neurons with connected to a single feature. So after the model is trained, you can take those weights and understand what kind of what is the individual importance for every feature. You can do it using various techniques. And one important question to ask yourself when looking at those biological aging clocks and when developing biomarkers of aging is, what does it measure? So one of the very simple biomarkers of aging is the person's face. So you can just use photographic markers to predict the person's chronological age. And a good doctor would even be able to tell you some guesses about your health status, about the patient's health status. And if the doctor, who is trained on several thousand patients, or maybe sometimes tens of thousands of patients, by just looking at them, can do this, it means that the deep neural net, which is trained on millions and never forgets, can likely do it better. So when we can see that for predicting the chronological age of the patient, the deep neural net can outperform a human. So for example, our deep neural networks always outperform me. But when you're training or when you're predicting the age using epigenetic data, we actually don't know what this clock measures and how does the damage associated with this clock accumulates, and how does it, how can we use it to develop all kinds of interventions, and what kind of interventions should be measured with this clock. And one way to evaluate those clocks is to compare them against each other. So here you see a comparison done by Dan Belsky at Duke University, one of the kind of top arbiters of biological aging clocks. And he showed that there is very little correlation between the various clocks. Even the epigenetic aging clocks developed using varying sets of methylation sites, they differ in performance on the same data set. And there is huge difference with other aging clocks, like physiological aging clocks. So those aging clocks can actually be also very useful for evaluating the value of the data type, for measuring the effects of your intervention, and to see where your drug or where your therapy works. Either it's a microbiomic level or transcriptomic level, tissue-specific transcriptomic level, or you can see a signal in the peripheral blood. So it's a very simple and cost-effective way to basically do data economics, understanding the value of the various data types and combination value of the variety of data types. So one of the very simple aging clocks that we developed using very, very simple data types coming from regular blood tests. So pretty much everybody has those features that come from either routine screening checks or a routine procedure when visiting a GP, your general practitioner. Those blood tests are cheap, very common, widely available, non-invasive, well, minimally invasive. So you just need a very simple blood test. They range from 10 features that you get, or even less, to a panel of 50 or more biomarkers, 50 like albumin, glucose, alkaline phosphatase, so something that the terms that most of us heard in school or got an interpretation for from our GP. And pretty much none of those features can be used as a good biomarker of aging by itself, or even in combination. So if you were to look at just maybe 10 features coming from your peripheral blood test, very few of them would correlate with your chronological age very well. Very difficult to train a predictor, and usually you get very low R, really low consistency for those features. But what we did in 2015 and published this paper in 2016, we trained an ensemble of deep neural nets trained to predict just one feature, age, on a very large number of samples, number of data sets, comprised of just 41 parameters, like, again, albumin, glucose, alkaline phosphatase, urea, cholesterol, some very, very simple markers in your peripheral blood. We trained 21 deep neural networks. Every single individual deep neural network actually outperformed pretty much every other machine learning technique and other statistical analysis techniques. But when you combine them into a stacking model using elastic net, you get much higher you get an improvement in accuracy. And we predict the patient's age reasonably well. Actually, patient's sex is predicted very well just by one deep neural net. And with an ensemble of the deep neural networks in 2016, again, those were very early days of deep learning. We achieved 5.5 mean absolute error for predicting the person's chronological age. And anecdotally, we started observing cases where people who are predicted to be older than their chronological age are also had some kind of age-related disease. And people who looked younger than their chronological age were healthy individuals, and most of them came from screening experiments. So there was anecdotal evidence that people who are predicted to be older, they actually are more sick. They have some kind of condition. We also, of course, compared all other machine learning techniques and demonstrated that deep neural networks outperform all other machine learning techniques and statistical techniques, even though the number of features in every sample is very, very little, very small. We still outperform linear regression by far with deep neural networks. And we used a technique called permutation feature importance analysis, where we construct a vector of all features going into the deep neural net and randomly shuffle just one. So, for example, randomly shuffle and albumin values going into the deep neural net. And then we look at how this random shuffling of that feature contributed to the accuracy of the predictor itself, of the entire kind of vector. And using this technique, you can create a histogram of importance of every individual feature for making that prediction. And it basically shows that albumin is one of the most important features in age prediction, and it correlates with liver function and with other tissue-specific changes in our body during aging. Of course, glucose is a common criminal, and its change in relation with other features contributes a lot to the accuracy of the prediction. So you can derive a lot of really interesting biological insights from those deep neural networks when you play with them. You can also build predictors for specific individual aging groups, for example, from 20 to 30, 30 to 40, 40 to 50, and see how those importance matrices, important histograms change for prediction of specific age, and how those features play in specific age groups. And using that technique, you can derive causal graphs. One other application of this technique of using deep neural networks trained on very simple data types in a very specific population was presented in our recent paper in Journal of Gerontology Series A, rather prominent journal in our field. There we used a very large data set. Many millions of samples were profiled, anonymized profiles were profiled to come up with a set of more or less healthy people within every population, Eastern Europeans, Canadians, and Koreans. And a deep neural network was trained for within every population on just 20 features to basically predict the patient's age. And then one feature, population group, was added, and we predict the population, the patient's age using a combined data set. And there were some very interesting, peculiar findings there. So one fun experiment to perform is to train the deep neural networks on one patient, one population group, and then test it on a completely different population group. So in this case, we actually trained the deep neural network on Canadians and got reasonably respectable mean absolute error, just over 5 years. And then we tested this predictor on Koreans, and we got higher mean absolute errors in Koreans, and the Koreans in general looked younger to the deep neural nets trained on Canadians. And when you test them on Eastern Europeans, they actually look older to the deep neural net, and as you can see, the mean absolute error increases. So it's a very fun way to demonstrate population specificity of the various data types and various biomarkers. So if you were to conduct a clinical study, it's a good idea to do an biological age adjustment on this level and on possibly many other data types. And that might be because, you know, in Eastern Europe, the equipment is different, the quality of those lab tests are less consistent, and or possibly because people are smoking and drinking, so we don't know, and it's a very interesting area of research. I will also skip a few slides just for the timing purposes here, but those slides are available for the viewership of this webcast. And another peculiar and interesting finding from this study was that some of those features that contribute most to the accuracy of the prediction are universal, like albumin, gender, well, sex, glucose, hemoglobin, urea. And some are more population specific, like triglycerides, HDL cholesterol, LDL cholesterol for Eastern Europeans. And you can go into specific population groups and start theorizing why those features are more important than one population than on the other. Maybe it's dietary, maybe it's genetic, and develop all kinds of theories using those aging clocks. So those aging clocks are hugely valuable for understanding the biology and also population sensitivity of the various data types. Another interesting finding from this particular study was that people who were, we tested those predictors on an independent data set called NHANES. It's a big longitudinal data set in the US where a variety of biomarkers are measured longitudinally. And you actually have links to some outcomes. So you can see how many people with certain markers died and what was the cause of death. So we tested our predictors on enhanced data set and got some really peculiar and interesting results. So people who were predicted to be 5 years or older than their chronological age using the predictor, on average, had much higher hazard ratio than people who were pretty much in line with their chronological age. And people who were predicted to be 5 years or more younger than their chronological age, they had much lower kind of all-cause mortality rate than people who were predicted to be within their age and chronological age. So that's a very interesting finding, and we can use it for all kinds of purposes to build better actuarial models or find ways to prevent all kinds of negative events and possibly reduce all-cause mortality or specific cause mortality using those clocks. And again, hematological clocks might not be the best clocks to do this. We think that transcriptomic clocks are much better and proteomics clocks are the best. But of course, if you combine those multiple data types, you get even more valuable data sets. So again, it will help us with data economics work. Another interesting study that we recently concluded and published was predictors of age using transcriptomic data coming from muscle tissue of the patients to study the process called sarcopenia, age-associated muscle wasting. And we generated and assembled a very large data set of muscle tissue transcriptomes from patients from 19 to 89 years old, and build a variety of predictors of chronological age using those data sets, using a variety of techniques like support vector machine, elastic net, random forest, and of course deep neural networks using a technique called deep feature selection. We also trained on signaling pathways to understand and get some biological interpretation of those predictors. And then we use the board account to kind of come up with the most important targets and most important features from all of those machine learning techniques to come up with the final ranks, and possibly identifying some targets that are implicated in that condition. So again, deep neural networks pretty much outperformed every other method, and we got to pretty respectable mean absolute errors with reasonably good consistency. We also used a variety of feature importance analysis techniques, and here we used deep feature selection techniques to identify the relative importance of every feature that we're feeding into the deep neural net. And using a variety of machine learning techniques, we ranked the various genes by their importance for predicting the chronological age of the patient. And you can see that various machine learning techniques give you very different scores for the genes that you're feeding into them. And for example, SCAP2 would not have been prioritized by deep neural nets, but it's prioritized by random forest, elastic net, and it's basically within the top 10 using various SVM techniques. But if you do the board count, it ranks number 1. And most of the kind of most important genes appear in the top 100 using pretty much every technique. And we also showed that some of those genes that are implicated in muscle wasting using this technique also are targetable, so they're druggable. And some of the genes, some of the possible targets, like, for example, the CA4 is implicated in muscle wasting also indirectly. So it's developed for the molecule that is targeting this particular target protein was developed for a set of other conditions, but anecdotally and in literature, it looks like people are using it and implicating it in all kinds of myopathies and myotonias. So it's indirectly and anecdotally associated with muscle wasting already. So it's we think that this research area is very promising, and we encourage people to go into this area and also collaborate with us. Another interesting technique, as I've mentioned in the very beginning of my talk, is generative adversarial networks. So GANs can be used for a variety of applications in healthcare. So again, generating synthetic data, but also generating novel chemistry. And usually it's a very good technique to employ and master, because if you want to validate some of those targets that you come up with using aging clocks or disease-specific clocks or biomarkers, you want to very quickly synthesize the molecules that hit those specific targets and not the others, and test those molecules in the biological system to do basically target validation. So to do that, we set on a journey in 2015, 2016, using all kinds of theoretical approaches to generate useful molecules using deep neural networks, using GANs, and spent basically 2 years in theory publishing a variety of publications. So one was the cornucopia of meaningful leads, possibly likely the first peer-reviewed paper in that field, and then followed with a series of other publications. And then in 2017, in mid-2017, we decided to start synthesizing those molecules that we come up with that have those properties that we want those molecules to have from the very beginning of the experiment. want to synthesize those molecules with those properties and test them. So we did it with Wuxi AppTech in China, and our first experiments resulted in failure. So again, in molecules, it's not as easy to generate very meaningful results and quickly validate. So when you're doing it with pictures, you can very quickly validate with your own sensory organs. With molecules, you need to synthesize and test, and sometimes the assay is wrong, or your molecule is bad, and sometimes you just don't know what happened. So our first experiment didn't result in a success. Then we had several iterations of this process and generated a lot of really cool proprietary data that we incorporated back into the generators. And around March 2018, we had a mild success. So most of the generation conditions that we've embedded in the molecule were met experimentally. And we used pretty trivial targets, very well-known targets like JAK kinases, EGFR, BRAF kinases as targets for the molecules that we're generating using AI. And we developed pretty respectable molecules that inhabited JAK1 and not JAK2, even though those targets are very similar, and JAK3 and not JAK2, and showed that we can now create very selective inhibitors for those kinase targets. But also those inhibitors carry many other properties that we want them to have, like solubility, bioavailability, log-SP, log-SW, blood-brain barrier, and basically up to 33 properties from scratch. And we developed some really cool BRAF and EGFR inhibitors that basically molecule that targets both proteins, and we can inhibit BRAF at picomolar concentration, and at the same time EGFR at, sorry, the other way around. So EGFR at picomolar concentration and BRAF at single-digit nanomolar concentration. So those are very promising results, and this molecule doesn't seem to inhibit many other kinases. And this is how we validate it. So we create the molecules, we train GANs to generate molecules with a specific set of properties. So in this case, you want activity against specific kinases. You sample from the latent space to see what kind of molecular structures are most likely to inhibit JAK1, JAK2, JAK3, or specific kinases. Then you synthesize those molecules and test. And in our case, generation conditions were met in more than 70% of the cases. And we identified very selective JAK1, JAK3 inhibitors, and published a variety of these experiments and also these models. So one of the really good papers to take a look at is the entangled conditional adversarial autoencoder for de novo drug discovery. It implements the new kind of disentanglement technique and manifold learning in the autoencoder, the adversarial autoencoder. And here we use SMILES representation of the molecular structure, so a string-based representation of molecular structure. And most of the groups can replicate this experiment. But most interesting factoid about this particular paper is that we actually synthesized the novel JAK3 inhibitor and tested it in an enzymatic assay and also in a biological assay. And we demonstrated that it can inhibit JAK3 at pretty much single-digit micromolar concentration. So it's not great, but not bad either. But it inhibits JAK2 at millimolar concentration, which is basically significantly larger than for JAK3, and it doesn't seem to hit other kinases strongly. So it might be a very, very useful molecule to test. And now we're doing this for many, many targets that we've identified using aging research and specific disease research. And we are licensing those molecules to a variety of smaller biotechnology companies and also to a large pharmaceutical company. And take participation in the future of this molecule. So we pretty much get paid only if it's successful. but we also do it a lot for academic purposes. Here is my contact information. You can reach us if you have any questions from the academic standpoint or from commercial standpoint. We are global. We have R&D resources in 9 countries, physical offices in 4 countries, and next year in 2019, we're moving into China in a very big way to work with the local partners there and to kickstart the pharmaceutical industry using AI. So we're very collaboratively, massively collaborative. Currently, we take on primarily only with very interesting projects. And if you're interested in learning more, please just go to PubMed and enter Insilico plus medicine. Thank you very much for your attention today.
Thank you, Dr. Zhavoronkov, for that informative presentation. I would like to also thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 2019. And as a final reminder, any questions submitted will be followed up via email by our speaker. That's all for now. Thank you for joining us, and we'll see you next time. Goodbye.